Okay, without further ado, let's move to the next session. The topic is biofluidic in post-harvest and cold change, cutting food loss and climate footprint. From Professor Dr. Bart Nicolai from KU Leuven, Belgium. This session will be led by our moderator, a lecturer from Faculty of Agricultural Technology, Ms. Hilda Maya Cynthia Dewi, MSc. As we begin, let's take a moment to introduce the moderator and her background in the field. Ms. Hilda Maya Cynthia Dewi, MSc, is a lecturer at the Department of Agricultural and Biosystem Engineering, Universitas Gadjah Mada, with strong academic background and in fresh produced post-harvest engineering, extraction, and process optimization. She earned her bachelor from Universitas Gadjah Mada and master's degree from National Pingtung University of Science and Technology, Taiwan. She has led and contributed to various research projects and publications related to post-harvest handling, microclimate effects on product quality, and value-added processing of agricultural commodities. Without further ado, please welcome our moderator in this session, Ms. Hilda Maya Cynthia Dewi, MSc. The time is yours.
Thank you. Good afternoon, everyone. A very welcome to all the participants here joining us today, both online and on-site. Welcome to this afternoon session of the short course in Agri-Food sector, organized by the Faculty of Agricultural Technology, Universitas Gadjah Mada. My name is Hilda. I'm very pleased to be your moderator for these sessions. It is a pleasure to see you all today, whether you are joining us in person or virtually. And today we are honored to have Prof. Bart Nicolai from KU Leuven, Belgium, to us. Okay, right now let's start the session with show the CV of the speakers. Prof. Bart Nicolai completed his doctoral degree at Catholic University Leuven, Belgium, and his research focuses on post-harvest biology and technology, as well as mathematical modeling. His work contributed greatly to the advancement of post-harvest science, particularly in the areas of fruit quality, storage system, low oxygen stress, and data-driven approaches in agricultural food research. Okay, and in today's session, Prof. Bart will deliver a lecture on biofluidics in post-harvest and cold chain. In this lecture, he will discuss different paradigm for modeling post-harvest processes, explain the distinction between statistical and mechanistic modeling, and also show how modeling can be used to analyze and improve post-harvest and refrigeration process. Thank you very much for coming all the way from Belgium to join us in this short course. We truly appreciate your presence, especially amidst these recent global uncertainties. Your willingness with us means a lot to us. And we are very grateful for your time being here today with us. So, ladies and gentlemen, please join me in warming welcome for Prof. Bart Nicolai. And before I hand over the floor, I would like to briefly share the format for today's session to Prof. Bart. For this session, we have allocated 70 minutes for your presentation, followed by 35 minutes Q&A session. And for all participants, both on-site and online, I kindly ask you to keep your microphone muted during the presentations unless you are invited to speak. And also when we move to a discussion session, you're welcome to raise your hand or send your questions through the chat box. We would appreciate if the question could be keep it brief and also relevant so that we can hear from many participants as possible. Yeah. With that, Professor Bart, the floor is yours.
Okay, thank you. Also, thanks to the organizing committee for having me here. It's not my first time in Indonesia, but it's definitely my first time in Yogyakarta. And just to maybe put the scene, this is a chart of Europe. And as you can see, there's this little red country here, which is pretty much in the center of Europe, and that's Belgium. And if you didn't know where Belgium is, the Netherlands is actually here. I'm guessing that the Netherlands is more known to you. This is France, this is Germany, Switzerland, Austria, and so on. And just see whether this works. Pointer does not seem to work for some reason. No. Maybe it works in the other window. Yeah, okay, now it's working. Yeah, okay. I think I shared the wrong window for some reason. Let me just try to fix this. No, this is not working. Can you maybe just show me what I have to do? Which screen I have to share, because I have a PowerPoint.
I think the correct one, but you need to.
I think we have two presentations open. I think I probably have to close this one, and then I have to share, I think. I have to share.
Then we can start the presentation afterwards. And that one.
Okay, perfect. Okay, so well, we are famous for a few products, but the ones that we are most famous for, and okay, here it is. It's Belgian chocolate. So these are like small pieces of chocolate, and there's always a kind of a filling in it. And if tourists come to Belgium, they always buy some chocolates to bring to their beloved wines. But we're also quite famous for our fruit and vegetables. And Belgium, although it's not as warm as in Indonesia, we are in a very favorable climate to produce many vegetables. Actually, quite a lot of them are produced in greenhouses, and together with the Netherlands, we have the highest yield in the world for vegetables such as tomatoes and so on, in a high-intensity production system. And this is really what my lab is about. So I'm a post-harvest technologist, which means that we study in my lab processes, all kinds of physiological and biochemical processes that go on in fruit and vegetables just after they are harvested. And based on that knowledge, we try to develop novel technologies to improve the quality of the products, and especially to maintain the quality after harvest as much as possible. So it's a little bit of a combination of physiology, a lot of engineering, and also modeling, because we try to model everything that we observe, and we use these models for optimizing process, a little bit in the same way as today an airplane or a space rocket is made. It's not by building all kinds of prototypes. No, okay, they design the car or the space rocket or the airplane on the computer, and then they do a few tests with only a few prototypes, and off they go. Okay, so that's the concept that we have in mind. We develop models for post-harvest processes. We use these models for optimizing the processes, for coming up with novel technologies. And so really models is behind much of what we do. And I know that this is kind of a mixed audience, with some of people that don't have too much background. But I will try to make it as simple as possible so that at least those of you that had at least some kind of basic mathematics, think about algebraic equations and so on, to still follow what I'm talking about. All right, so the title of my talk is Biofluidics in Post-Harvest and the Cold Chain. Biofluidics basically means transport of all kinds of fluidic processes, but fluids is now very general, meaning also transport of gases. And in fact, the term biofluidics also incorporates transfer of heat. And what is essential to all kinds of post-harvest processes, in general to food processes, is what is called transport phenomena. Transport phenomena is transport of heat, so heating of stuff or cooling of stuff, transport of momentum, which means flowing, stuff that is flowing because there's like a fan or there's a pump that flows the fluid, but also transport of gases. Think about transport of oxygen through plastic materials and things like that. And I'm going to take a kind of a modeling point of view, and I'm going to show you how you can use this to improve post-harvest processes in such a way that you can cut food losses. And I will also show you how you can do these calculations to kind of optimize your storage system. And if you do so, you can also save some energy. All right. So this is pretty much kind of a talk about many of the research topics that we have in my lab that have been executed or are being executed in the past by many of my PhD students. So what I want to do today is I'm going to compare different paradigms for modeling post-harvest processes. And paradigms, I don't know whether you know the word, but it's just like a fancy term for concepts or strategies. Okay, so I'm going to discuss those different strategies. I will definitely focus on the difference between statistical and mechanistic modeling, and I will show you that one is just trying to fit data with some simple models without any physics background, and mechanistic models is then it's the inverse, it's the opposite one. It's basically trying to include much physics and biology and chemistry knowledge as possible and try to translate this into mathematical equations. And then I'm going to give some examples to show you how you can actually use models in your research or in your practice as a post-harvest engineer. But it's going to be more broader than that. There's a little bit of lag between what is happening on my screen and what is displayed, but I will do my best here. So this is basically what I'm going to talk about. I'm going to talk about what modeling is. Then I'm going to talk about statistical models. Going to talk about a little bit about artificial intelligence and deep learning, tell you what the basic is and explain to you that it is nothing else as a kind of a statistical model. Then I'm going to talk about kinetic models, which are models that can describe things that, or variables that evolve as a function of time. Think about the temperature at one particular point in this auditorium. It's something that changes. Maybe now it's kind of coldish because there's an airco, but if we would turn the airco off, then the temperature is going up. So it's a variable that changes as a function of time. So these are kinetic models. Then I'm going to talk about physical models. Physical models are models that really incorporate much of the basic physics that describe transport phenomena, that describe transport of heat, mass and momentum. These are the same models that are, for example, used to do predictions of the weather of tomorrow and the next couple of days. So really fundamental physics-based models. Going to illustrate this based on some research that we did on gas transport in pear fruit during a very particular type of storage that is controlled atmosphere storage. I will explain what it means by then. Then I'm going to move on to computational fluid dynamics, and you can already expect from the word what it means. It really means using these fundamental models of heat and mass transfer to do long-term predictions of all kinds of flow problems in a kind of an enclosed environment. For example, think about the climate here. If we would turn off the air conditioning, then the velocity profile, because if the air conditioning is on, there's air that is flowing all the time in all kinds of different directions. If we turn it off, then there's no airflow anymore. The temperature is going to rise. Okay, so many other things. The carbon dioxide that we are all exhaling is going to stay in place. It's going to only slowly move, whereas otherwise it is taken by the airflow. Well, this, in order to predict this, you are going to use a technique that is called computational fluid dynamics. So that's the next agenda. Then I'm going to talk about discrete element models, which is models of individual particles that are interacting with each other, such as, for example, fruit that are shaken when they are transported on a truck. And then in the end, I'm going to shortly talk about integrated modeling environments, which are kind of software packages where you can play, and there is a model for a refrigeration plant. There's a model for the fruit in it. There's maybe a model for the truck that you use to transport your fruits and so on. And in this way, you can analyze the whole chain from the production, the time of production, so basically the harvest of the fruit, until the very end of the chain, which is when the fruit or the vegetables end up in the shop. Okay. So I'm going to make it as simple as possible. If you have to talk about modeling, there's no way that you can avoid equations. I'm going to really use a minimal amount of equations, and then I'm going to focus on what they mean, not on what the individual meaning is, because there's no time to explain that all in detail, but I would like to at least convey the concepts to all of you. All right, so let me first start with some basic stuff. A model is a kind of an abstract representation of reality, okay? It is concise, okay? One model of a table, for example, with lots of numbers, would be to have the table as the model, okay? It's perfect as a model, but it's a very inefficient model because there's no data compression. So typically with a model, you want to have a concise description of what is in the data, not just presenting the data themselves, okay? So there's always kind of a reductionist step in it, which has some consequences. Often it builds upon mathematics or logic, but that's not necessary. We just, for engineering purposes, it is kind of handy to have a mathematical representation because we can use our whole big toolbox of mathematics to basically deal with the models and to solve them. But there are some models that are not mathematical. I will give some very simple and very useful examples that are also very fine. It all depends on what you want to do with the model. Then the last one is also very much, the complexity must match the objectives, okay? There was once this guy, Albert Einstein, that most of you will know. He did some very important developments in physics. And he once told you that the model should be as simple as possible, but not more simple than that, okay? So sometimes you can have very, very simple models and they will be perfect, but sometimes you want to do something kind of complicated. Well, then you need to have like a complicated model. Okay, so this is all kind of very abstract. What does it mean? Well, for example, this is, well, a very known phrase of Albert Einstein. I got it from the internet. A model, this is just the original picture. I'm going to make a model of that. Actually, I used AI to make a very simple model of this. And the very simple model that I have is this one here, okay? So you can still recognize the basic features in the sense that there's a guy with, well, it's a person because he has two eyes and he has a nose and he has hair, a little bit of a wild version of it. And then he has a mustache and he also has kind of his tongue that is sticking out of his mouth. So while we all see that this is definitely a simplification of this image here, there's only three levels of color. That's what I asked the AI to do. And at the same time, the essential part of it, I think, which is that he is sticking out his tongue, is maintained. Okay, so this is a kind of a model of smiling Albert Einstein. And most of the features are kind of maintained. But of course, there's also some information that is lost. For example, the shape of his ears, the fact that he has pupils in his eyes and so on. They are lost, but we don't care because we know that everybody has pupils in his eye, or most people have pupils in their eyes. So that's all fine. So this is a kind of a model of this image of Albert Einstein. But we could say, well, actually, the only thing that I want to convey is I'm writing like a little chat, and I want to indicate that to convey some message, a message that you convey if you stick out your tongue. And I can just use an icon like this one here, okay, where you have like a smiley, but then with a person sticking out his tongue, okay? So this is pretty much the most simple translation of this complicated picture into something that is meaningful. So this is a model. And it depends on the purpose of what you do. If you want to say something that for which you need an icon of a guy sticking out his tongue, and you want to introduce that in your chat, this is a perfect model for this, okay? But maybe if you want to really show all the details of Einstein's face and whatever and so on, then of course you have to go to the original model. So this is what I mean, that typically a model is going to be concise, but the level of complexity depends on what you want to do, okay? You don't need to make a model very, very complicated if you just want to convey a very simple message. That would be really stupid or a loss of time, unless you like to make models. Okay. So why do we model? Well, we can model to analyze experimental data. So if you do any analysis, or most statistical analysis that you do are based on models, even if you don't realize that. So I'm assuming that most of you have had some kind of a basic statistics course, then you know what a regression model is. You know what an ANOVA is. A regression model is, of course, explicitly a model, but an ANOVA is also making a model. You don't think about creating a model, but you can actually prove that making ANOVA is perfectly equivalent to making a regression model, a generalized model, actually. Okay, so you also make a model there. So you basically use models to analyze experimental data to find out trends or to kind of analyze hypotheses that you made before. You can also want to predict the future. For example, you can use very complicated weather models, like some of the ones that I'm going to discuss later on, to predict whether tomorrow is going to be sunny or not. Well, here, I have a very simple model that I can already use, okay? Once that happens, that is often applicable, that's the weather of tomorrow is the same as the weather of today. Okay, that's a very good model, and it's going to work here even better. Okay, so I don't need very complicated equations. This model is more than good enough for many practices and so on. But sometimes you don't know that. If you live in Belgium, for example, you don't know the weather, what's the weather going to be tomorrow. Or sometimes you want to predict something. For example, you want to predict whether the stock, suppose that you have shares of a certain company, you have shares of NVIDIA, and you want to know whether you have to solve these shares and make a lot of profit, or you want to keep them for one more month. So then you would like to make a prediction. And for that, you are going to use a model, okay, of a certain complexity. You can also use it, and that's what we do in my lab, and what many of you that are doing like an engineering degree will do in the future. That's the design processes and equipment. You can also use models to understand nature. I'm going to give a very simple example of that later on. And the one that often drives my research is to reveal beauty, the beauty that is embedded in biological systems, in the way how that biology works, the biochemistry, and so on. So these are all good enough reasons, but it always depends on what you want to do, and the complexity of the model really depends on what you want to do. If you want to understand nature or reveal beauty, then typically you would need to have more complex models than the simple ones. Okay, well, again, it's still very abstract. Let me give you a few simple examples and show you the fact that there are models everywhere, that we're using them all the time. A very simple model is, and now I'm in my area, which is fruit and vegetables. This apple looks green, so it must be firm and crispy. So in Belgium, we have this idea that if you have like green apples, that they should be very, very crisp. And if they are yellow, then they are soft, okay? There's a very good reason for that, and that's because apples, if they age, the green in the apple skin is caused by chlorophyll. And chlorophyll during ripening is broken down. And if the chlorophyll is gone, then there's a left color that is left. But at the same time, the apple becomes softer. Okay, so this happens, by the way, with many, many fruit, also with kiwi fruit or whatever, or with banana fruit, for example. So this model says that I just have to look at the apple and then I have to observe whether it is green or yellow, and this will already tell me something about the ripeness and hence the firmness of the apple. Okay, this is this is a model that you would, well, know that there are not too many apples here, but if you go to Europe and you go to a shop where you would buy an apple, any consumer will have this model, and he or she would look at the apples and would see, well, they're yellow, they're going to be soft, will not be able to keep them long, so I'm not going to buy them. Okay, so it's a model, it's a verbal model. You can also have logical expressions, and this is something that comes from computer languages. There are special computer languages to describe logic. It says that if color of the apple in between brackets is green, then texture of the apple is firm. Okay, so this is how you would basically code this in logical programming systems. But what we are, or what I'm going to talk about most is mathematical models. And mathematical models have some sort of a mathematical relationship between the variables. And here you can see this model that I'm going to use. It's just a simple equation. The firmness is proportional to the color. This is a measure of the color. It's the A star value. If you don't know what it is, doesn't mean, but it's a measure of the redness of the apple or the greenness of the apple. So, and then you have two parameters in it, okay? You can see that there's still this relationship between the firmness of the apple and between its color, but now we have a kind of a concise mathematical representation, which also includes two numbers, two specific coefficients of the model, okay? If I want to use this model, I will need to determine the value of these properties, okay? This will require some effort because for this I will need to do measurements, put the data in a table, do a statistical analysis, and then the statistical package will give me these numbers. But the award that I have by doing so, that this will make it able to make precise predictions of how firm the apple will be in the future, okay? So you can see there is a kind of a next level of precision of these models. These models tell us much more. And if you know, for example, that the green apple or that a firm apple is like more expensive, will give you more money if you sell it than a red or a yellow apple, then with this model, you can actually predict what your income is going to be. Okay, so this is really next level. Okay, so this is why in any engineering curriculum or a science curriculum, we teach also mathematical models, because for certain purposes, they are better than other models. But as I mentioned before, it depends on the purpose. Okay, so which are the advantages of mathematical models? Well, it enforces you to think critically and organize your thoughts. If you write up a model, you have to think about all the components of the model, so you really have to understand your process. The second is that it can be used to design experiments and reduce work. There's a whole theory of experimental design in statistics. Our students of the third bachelor's years get such a course. And in this way, they are going to use simple statistical models to reduce their workload. Okay, if you do experimental design, it means that you can get the same information with an experiment that will only take 5 days instead of 10 days. So that's a big advantage what you can get. Therefore, you can use models. You can make it for quantitative predictions, as I just said before. And today, modeling is a part of many curricula, especially if you are into biology, because in biology now you have all those what they call omics techniques, which means that you are doing transcriptomics, genomics, metabolomics, lipidomics, or whatever, proteomics. And the data that you get from them are really huge. Okay, so there's no way you can look at those data because it's like a million numbers. You have to analyze them with appropriate software, and behind this analysis, again, there are models. Okay, so there's not really an option to avoid it in the science of today if you are into biology science or engineering or chemistry or whatever. Okay, so this is my introduction. I'm going to talk about two approaches. One are empirical or statistical models, which are models that basically lead us back to our basic toolbox of mathematics, where we learned about functions, about relationships between a y variable and an x variable, and then you can do this with a simple equation, this relationship, as I did when I was discussing firmness and color, or more complicated models. But these models, they have no physical meaning. It's just an equation, an abstract equation, but we just use it to describe the data to make our life a little bit easier. The other type of models are mechanistic models, and they build upon the underlying physical and chemical laws. And they have certain advantages and disadvantages because there's physics built in, which inherently means that they have more power and also more extraplanetary power. You can use them to predict things for which they are not trained. Okay, this is in contrast with, for example, more statistical models such as neural network models, where they can produce nonsense. We call them then hallucinations, okay, but they don't mean anything. They're not physical. Okay, let's start. First type of models are simple, what they call univariate regression models. So these are the basic models that you use to make calibration curves in a lab and so on, to make simple predictions. And they just give a relationship between some response variable y and some explanatory variable x. And there are a few coefficients in them. And typically, the way how you estimate these coefficients is by a method that is called least squares regression. You even don't need to know how it works, because you can go even into Excel and you can actually calculate the regression coefficients very easily. You just have to provide the measurement data of different values of y that correspond to different values of x, and then Excel will basically produce the coefficients of the model. For many purposes, this is good enough, but the prediction value outside the calibration or the training range, because training is another more modern word for calibration. If you do that, then you can make huge mistakes, and it does not lead to any insight. So we can see that there is a relationship. If the fruit goes from green to yellow, it becomes less yellow, but it becomes also less firm. We can use the color measurement to predict the firmness, but we have no idea what the underlying reason is for it. The underlying reason is that the fruit ripens, and that there are many processes that are coordinated and that kind of many parameters that change. But the equation itself doesn't provide us any understanding for that. You can also kind of further extend this. Well, this is, before saying this is like a graphical representation of such a linear relationship. You can see that the firmness goes down. as a function of time. Time is actually in this case, my x variable. So these are functions like this. You can fit a straight line through these data. With Excel, you can visualize it and so on. And for many purposes, this is good enough. But often the number of explanatory variables is much larger. For example, firmness of the fruit can depend on the color of the fruit. It can depend on the time. It can also depend on the humidity of the room. If the room humidity changes, the firmness will also change. It can depend on maybe the cultivar of apple and so on, which means that you have more explanatory variable. And that's fine because you can extend this simple linear regression model that I showed you to multiple dimensions, and then you get a multiple linear regression model. And you can also deal with that easily in Excel. Okay, Excel doesn't know, you just have to provide different columns for the different variables and do all kinds of interesting things. But again, it allows you to describe more complex data, but essentially it's a simple statistical model. Another model is a neural network, and a neural network is really at the heart of artificial intelligence. And how does that work? Well, instead of like a simple calculation where you have like one variable x that is transformed in a variable y, you have actually many different computational nodes, and in every node, such a simple calculation is done. So there's an input, and then there's a corresponding output that comes out of this node. And but all those nodes are organized in a kind of a network topology, and this mimics a little bit how the computational nodes that are called neurons in our brain are connected to each other. And the whole idea of such a network is that you have like different inputs, you have different outputs, but there are also kind of intermediate layers that are called hidden layers. So we don't have a direct relationship between the output variable y and the input variable x, or input variables x or output variables y, but they are processed in between. And what happens is that at every node, the software will basically calculate the output of this node, this node, this node, and this node, but multiplied with a certain weight. Okay, so this output is multiplied with a weight of 5, this one with 1, this with 0.1. So this means that this node is going to be very important in determining what is going into this node. This node may be a little bit less important, and so on. And in this way, all those nodes are calculating all those things with all those inputs they get from previous nodes, and then they provide an output. Now, how does this help us? Well, remember that in a linear regression model, you have two coefficients that you need to determine. Okay, here the nodes' weights are the variables that you need to determine. So these are coefficients. And for every of these arrows, you have a number that you have to estimate, basically. So what you essentially do is, like, if you do a linear regression model, you have X data and you have Y data, and you ask Excel to determine the coefficients of this relationship. Here you have X data, you have Y data, and you ask your network software to basically use these data and estimate those weights, the value of those weights that gives you the best relationship between the X values and the Y values. So it's nothing else than a regression model. The only difference is that you have this typical network topology, which is different than normal, and you have lots of data, okay? But all neural networks work in this way. So if you take ChatGPT, for example, or Claude or whatever, these are all neural networks, and they are essentially all regression models. Okay, it's not more simple than that. What is different is that they have underlying complexities, different layers that do all kinds of other calculations and so on. But if you ask a question to ChatGPT, it takes the inputs, and the inputs are the words that you basically ask, and then ChatGPT predicts the output, and the output is its response. Okay, but it's a kind of a complex network because it has 30 trillion parameters. Okay, compared to our simple regression model that only had like 2 parameters. But it's a regression model. Okay, but a very useful one. Okay. But I'm really, well, we also do a lot of neural network model, artificial intelligence in my lab, but I'm really mainly interested in more mechanistic models that have like a firm basis in science, in biology, in chemistry, and also in physics. And I'm going to start with the most simple type of models, which are kinetic models. And in this case, the fruit or the vegetable is essentially considered as a chemical reaction. You know, these big chemical reactors here, which have all kinds of unit operations, many tubes in between them. And in such a reaction, in such a reactor, you can have thousands of reactions that happen at the same time. And this is exactly the same as in our cells. We have many biochemical pathways that are running all the time. They are transforming glucose into carbon dioxide and so on. So it's basically a biochemical reactor. And there's a formalism in reactor engineering to model such reactors, and we are going to use the same formalism in basically describing what is happening in fruit and vegetables. The only thing is that the language that we are going to use for this is a little more complicated. We are going to use, instead of simple algebraic equations like regression equations, we are going to use differential equations. And I know that this is a little bit of a mixed audience, but who knows what a differential equation is? So if you have had some basic physics background, then you should at least know. Nobody says so. Maybe I have to revert the question. Who doesn't know what a differential equation is? Okay. Who is not sure whether he or she knows what a differential equation is? Okay, at least a few that are not sure. I'm wondering what those of you that don't know what a differential equation is and also don't know whether they know it or they're not sure what they think about it. Okay, I'm not going to explore that. Anyway, a differential equation is an equation that involves the derivative of a function. Okay, you know what a derivative is. Perfect. So I'm assuming, because you must have had this in your undergraduate mathematics, in your secondary school, your high school mathematics, okay? And a differential equation is a relationship between the derivative of a function with other variables, okay? And then you would say, hey, this looks like very simple, but consider like this, you have the velocity. The velocity is how much meters you do per second. So it's basically distance divided by time, okay? But the thing is that I can kind of go slow or I can go rapid and so on. So my distance is changing all the time. So the velocity, you can better define it as a variable that at one arbitrary time gives me the instantaneous velocity, okay? And the instantaneous velocity is then the change that I make, a very, very small change that I make in a very small time instance. And something very small change as a function of a very small time is essentially the derivative of that function, okay? It is a function of time, because now I'm going fast and now I'm going slow. So the derivative is a function of time. And all the laws of physics are represented basically as a function of a differential equation. And the most famous one is actually the Newton's law that gives you an equation that allows you or that gives you a relationship between some derivatives, actually second order derivatives, but that's not important, and the force that is applied. And you can solve this differential equation. And many of you must have done that in your undergraduate physics courses to predict the time course, the motion of an object when there are forces working on the object. Okay, so this is the language of nature, it's differential equations. Okay, I'm going to try to explain it because the challenge now for me has been increased a lot if you don't know differential equations. Okay, so this is my differential equation here. Here I have like some component x, which can be, for example, let's say sugar. Okay, sugar content. So between brackets means the sugar concentration. So the sugar concentration is going to change. So this is the differential or the derivative of the sugar concentration with respect to time. So this is a derivative, okay? And it says that this change depends on some function that is a function of the sugar concentration and maybe of another variable. Okay, this is still abstract, but I will make it more concrete in the next slides. And then there's maybe another component, which is the carbon dioxide concentration. But we know that the sugars are transformed into carbon dioxide because of respiration. Okay, so there's the second equation that describes me how carbon dioxide is going to evolve. That must also be a function of the sugar concentration, because if there's a lot of sugar, then I will basically convert a lot of sugar to carbon dioxide. So this must be definitely an effect. And maybe the carbon dioxide concentration itself has an effect on the change of the carbon dioxide concentration. So these are two differential equations. Okay. Let me just, maybe before making this a little bit more clear, the thing is then now, how does this function look like? So this is a function of the two variables of my system. And I'm just going to give you like one example here, which is so-called Michaelis-Menten enzyme kinetics. And for those of you who did not have like a biology background, an enzyme is a protein. So it's a compound in a cell that catalyzes one biochemical reaction. Okay. There are many of them. This is basically what drives our body, all kinds of enzymatic reactions. And enzymatic reactions they work typically like this. So you have like one substrate. Suppose that this is like sugar. Sugar binds to the enzyme E. So we have a kind of a complex. Some of it will go back because this is a reversible reaction. And then there's something might happen, and then basically this X might be transformed into another component. Y, this can be, for example, carbon dioxide, and the enzyme becomes free again to catalyze other reactions. So this is really a basic representation of an enzymatic reaction. Okay, now I'm going to translate this into a differential equation, and I'm not going to go into the details, but the typical shape of this differential equation looks like this here. The change of my sugar concentration in time is proportional to the amount of sugar, and that's kind of logical, because if there's more sugar to be transformed into carbon dioxide, well, there's going to be, if there's more substrate, then the reaction speed is going to go up because there's more sugar that can be transformed per unit time, okay? But because of this complexity of the reaction, this rate, the right-hand side of this equation is a little bit more complicated here. And if you translate this into a graph, then you can see this is the concentration of substrate, that at high concentrations of substrate, the reaction rate kind of levels off, which means that there's almost only so much enzyme. So there's a kind of an upper limit of how fast sugar can be converted to carbon dioxide. Think about our body, okay? We can eat a lot, okay? If you don't eat, there's no sugar in our body, then you don't have energy. You can only walk very, very slowly. But if you eat more, like that's what like cyclists and marathon runners do, if they eat more sugar, like they eat a banana, then they can actually run faster because they have the energy from the banana, okay? But they can eat one banana, but maybe you would say, okay, but if they eat one banana, they can run like 20 kilometers per hour, maybe give them 2 bananas, and then maybe they can run 40 kilometers per hour, and then why not give them 10 bananas? Okay? But it doesn't work like that. There's a kind of a leveling off. Okay, so this is what this means. At some point, the rate just levels off. It also means that when you give them, if they're really starved, and then even if you give them like a little cookie, even then they would be able to start running, maybe not a lot because it's a small cookie. So then you have to feed them more and so on. So this is this kind of complex relationship between the rate of a reaction and between the concentration. So that's basically embedded in this equation that I'm going to skip. Okay. So what are the advantages? Models like this, you can actually extend this to a model of a whole chemical reactor. And the advantage is that they're mechanistic, so they are based on physical laws, which means that they can predict also outside the range you develop these equations. There are some disadvantages. Well, of course, the mathematics is more complex, but there's also software for dealing with this. The parameter estimation is more difficult because you have more complex parameters. But then again, you just rely on existing software to solve this kind of problems. Okay, this I'm going to skip. This is too complex. So now I'm going to give you one example. This is the last example where we really have to look at equations, okay? But it really illustrates the point of how you can, starting with this simple machinery, to make much more complex relationships. And I'm going to talk about climacteric ripening. And climacteric ripening is when you have certain fruit, such as banana fruit, you pick them and you can pick them even unripe, and then they still would become ripe. This is called climacteric ripening. This is really great. Otherwise, if this would not exist, then bananas in Europe would be really expensive because there's no way you can grow bananas in Europe. Actually, you can do it. We grow bananas in Leuven, but that's in a greenhouse because we have an important banana center. But the way how they are dealt with is that bananas are picked unripe, green, and those that come from that arrive in Europe are typically come from Central America and from South America. They travel for 3 weeks, and because they're unripe, you can transport them and nothing happens. They stay green. Okay, they are kind of in a hibernation state. And then when they arrive in Antwerp, which is the main port of Belgium, it's the main banana port of Europe, then the bananas are put into ripening rooms and they treat them with ethylene, which is a very small or gas of very small molecules. And it's also a ripening hormone of fruits. And then all of a sudden, there's a treatment of 24 hours, bananas start to ripen. And within 7 days, they go from green to yellow with brown spots. Okay? And that's really a spectacular property of banana. It's not only banana that has this property. Also apple and pear and tomato and mango fruits, they all have these properties. Fruit that do not have it are all kinds of citrus fruits, for example. Pineapple is not climacteric and so on. But at least the large part of fruits, including both temperate and tropical fruits, are climacteric. So if you can kind of understand this ripening process and maybe predict it, then you can better organize your post-harvest processes. Because if you don't do it right, if you have bananas that are picked too late, what happens is that they're transported in a ship. Then if one banana is ripe, it will start producing ethylene gas a lot. and it will start to ripen the other bananas. And then before you know, you have a ship that is filled with yellow bananas and they have no commercial value anymore. You have to destroy them because it's too late. You cannot commercialize it. It's a huge loss. So here, maybe you want to have a model that is a little bit better than if the banana is green, then if it's not too green, then it will stay green. Otherwise, it will be yellow. You want to have kind of a quantitative model. Okay. And the models that are used are relatively simple regression models, but I think we should better go to more mechanistic models. And that's what I'm going to explain to you. So you'll have to bear with me to follow the next slides. So this is the time course of the oxygen consumption rate, the blue one here. You can see that in the beginning of the fruit, it's very unripe, but it needs a lot of oxygen because it still has to grow. Okay. And then it decreases, and at some point it gets into a kind of hibernation state. The oxygen consumption rate is constant. Sorry, this is the, okay, this must be ethylene, sorry, made a mistake here. So there's no ethylene, and then at a certain time, the ethylene production goes up, and also the oxygen consumption rate goes up. So this is kind of coordinated. And we wanted to describe this coordinated action. And I actually made kind of an overview of the different equations, but I'm not going to go too deep into it, but just give you an idea of the whole concept. So this is my pear fruit, and this is my simplification. So I'm not going to discuss all the different steps of the respiration metabolism because it's very technical, many enzymes involved and so on. I'm only going to consider like the respiration process as one biochemical reaction and the ethylene synthesis as another biochemical reaction. So that's a huge simplification, but as I mentioned before, that's what you do when you're modeling here. So oxygen diffuses into the fruit, then it is consumed for ethylene biosynthesis, because one of the steps of ethylene biosynthesis is that it uses oxygen. There's an oxidation reaction. Then oxygen is also used in the respiration process, and it produces, basically, both produce carbon dioxide. Carbon dioxide is transported out. Ethylene biosynthesis, of course, results in ethylene, and ethylene is also transported out of the fruit. So this is essentially what is going on. Okay, I'm just going to show you one step of the model, and then the other steps are very similar. If you're interested, you can look at the slides, just to give you a little bit of a feeling of how you develop a model. And here we have our differential equation. So the first one is for ethylene. It says that the change of the ethylene concentration in the fruit is a consequence of biosynthesis. So this is the biosynthesis term. You can see that you need oxygen. So oxygen is somewhere in the numerator here. The more oxygen, the faster the biosynthesis goes. And then it also is transported. So depending on whether the ethylene concentration outside is higher than the internal ethylene concentration or the other way around, you have transport over the skin of the fruit that either increases the ethylene concentration inside the fruit or decreases it. Now, there's also a rate here, and for the rate, we just assume that it is proportional to the concentration of both enzymes that are responsible for ethylene biosynthesis. Okay? And then, of course,
gentle reminder, we have less than 10 minutes remaining for the presentation.
We'll have to speed up a little bit. So you can see that then, of course, you can establish novel equations for these two enzymes and so on, and you can go to the RNA for those of you that are familiar with it and so on. I'm going to skip this. This is in my next slides. Okay. But you can see in the end that you have many equations. Just go back here. Many equations that are all differential equations, and they basically describe the whole dynamics of this system. And then in the end, you can, of course, you have to train the model. You have to find out what the parameter values are that I'm going to skip. And here you can see the end result, which was done by one of my PhD students, by Ming. And here, the red dots are the measurements and the blue lines are the model predictions that go through the measurements. Okay, and you can see that overall there's kind of a good agreement, so that made us quite happy. But there are some kind of differences that are systematic here. For example, the model always overpredicts the oxygen production rate and the ethylene production rate, and here the model underpredicts the oxygen rate. And this information is very useful because it means that the model is not capable of describing your data, which means that some of the biochemistry that is probably important has not been captured by the model. Okay, and this can lead to new research, okay, that you can then try to establish and refine the model. So we use these models to understand our understanding of the ethylene biosynthesis. Okay, next type of models that I'm going to describe are so-called physical models. And you might have recognized here the Orion spaceship here that a couple of days ago kind of ran down. And this is the heat shield of the Orion spaceship. So this was predicted by a physical model for heat transfer. And you can see that it's kind of hot. Red means hot. And that's because it's 3000 degrees. Okay, and then it cools down, and this is like a cooler stream at the end of the capsule and so on. And to describe these phenomena that depend both on space and time, you need much more powerful tools. And what we use for this are transport equations. And this is, for example, a transport equation for heat. And without going into details, you can see that there are also derivatives in this equation, but these are derivatives with respect to both time and with respect to the spatial coordinates. So more complicated transport equations. And again, also parameters, but some of these parameters are constants that you can find in tables and so on. So how can we use this information for post-harvest processes? Well, I'm going to show you how we applied it to the development of browning in pears. So this is a problem that we have in Belgium. If we store those pears under too low oxygen conditions, then the center will become brown. And this has something to do with the fact that the metabolism changes to a fermentation metabolism. Of course, pears that have this disorder do not have any commercial value anymore. So we wanted to understand what was going on. This is a typical view of these large cool stores that are maybe 3 times the size of this room, 3 times as high as this room. So if your pears are affected, it's a lot of pears that are affected. So it's a lot of loss. So I'm actually presenting some of the research of Bayou now. So this is our hypothesis. So if you have a low oxygen storage atmosphere, then the oxygen concentration inside the fruit is even lower because the little oxygen that gets in here is consumed. So the oxygen goes lower. And then the system changes to a fermentation metabolism. It starts to produce ethanol, which we don't want to have in fruit. And there's also an energy crisis because the energy yield is very, very low. So you will get all kinds of damages like membrane damage that can no longer be repaired. And then you have like phenolic components that are getting in touch with polyphenol oxidases, and you have browning and disruption of the tissue. So we looked at this for many times from many different perspectives, but here we focused on the gas transport. And if you use, let me just skip this. If you use these transport models, this is, for example, a simulation of the oxygen concentration inside the fruit and the carbon dioxide concentration in the fruit. Then you can see that, well, there's some change in blueness. This is dark blue and so on. This is lighter blue here. But here you can see in the carbon dioxide concentration that the carbon dioxide concentration in the center of the fruit is very, very large here and a little bit less here. And if you take the ratio of both of the carbon dioxide production and the oxygen consumption, then you would get a variable that we called RQ, which is a measure of the ratio of fermentation relative to respiration inside the fruit. So this is a totally computed result. And if you compare this to the shape of the disorder in brown, so these are computed tomography images. So the brown is the actual brown spot inside the fruit, and the blue is the computed one. You can see that there's not a perfect match, but there's a quite good agreement between these low oxygen profiles and the disorder. So here we used a mathematical model essentially to predict complicated aspects such as the browning internal disorders in fruit during post-harvest. And you can solve this, then you can basically reduce the number of lost fruit considerably. We also, excuse me. We also found out that size matters. The larger the fruit, the lower the oxygen concentration, blue means very low, and the higher the carbon dioxide concentration. This also corresponded that what we found out in reality, that big fruit are much more sensitive to this disorder. I'm going to skip a few slides here.
Yeah, Prof, but we have about 3 minutes left before the presentation.
Okay, so the last, or maybe I'm going to, the last formalism that I'm going to shortly describe is computational fluid dynamics. This is the same as before, but the models are even complex. And these are the models that are used for weather predictions, but also to design, for example, airplanes. You can see here the flow lines of air on the top of an aircraft wing and so on. But there's also special software that is used nowadays exclusively to design such phenomena. And we are trying to use these models to also optimize post-harvest processes. For example, here you can see a cool room where we calculated, this is the cooling machinery. It produces cold air that is blown to the front of the fridge that is filled with big boxes of fruit. You can see that the cold air flows through the stack and then returns to the cooling machine where it is cooled again, and this goes on and goes on. And here you can see the temperature inside the stack. You can see that there is a gradient of the temperature inside the cool room here, with the hottest temperature below the evaporator, because the air that gets here has to pass through the whole stack, and meanwhile it heats up. Okay, let me just show you some of the results. So what we for example, did was we tried to find out what happens if we have less layers of boxes in the cool rooms on top. How does this affect the flow pattern and the temperatures? Or if we remove some of the stacks here in front of the cool cell, so just under the evaporator. And then I'm not going to go into too much detail, but there's a little bit of a lag here. But here you can see, for example, what happens. This is where you remove both layers here and also a column here. And then it turns out that the air kind of circulates. Okay, so it doesn't penetrate the stack, so the stack cannot cool down anymore. And there's different combinations of rows that we removed and so on. I'm not going to discuss them all. But this means that the stacking pattern inside the cool room can make a big of difference. Okay, and you can also do some simple adjustments. For example, there was a cool room and the farmer complained that the temperature was not uniform in his stack. So he measured the temperature, and indeed here at the bottom, this is a kind of vertical cross-section, we found that the temperature was red, which was high. So we basically kind of put a little bit of a plastic thing here in between the cooling machine and between the upper stack, in such a way that the air could not kind of flow back beneath the evaporator between the cooling machine. So by doing this, we forced the air penetrating the product here. And in the end, we saw that the temperature was much more uniform here in this cell, so the temperature were much cooler, much less red than here. And this was a solution that cost the farmer 300 euro. Okay, and so it was like a good idea. I'm going to skip this. Yeah. And maybe just the last type of models here are discrete element models. Discrete element models go back to this example of the laws of Newton. So suppose that you have like fruit that are moving, for example, on a sorting line, or in a transport, they bump into each other and then you get damage and so on. And we wanted to predict this. You can, of course, send the truck from Belgium to Poland with fruit inside and then measure the quality of the trucks in the end, but we want to have some predictions so that we can play a little bit with the parameters. And the modeling type that we do is discrete element modeling. So basically, we consider every object as something that interacts with another object while they're falling, and we solve Newton's equations for motion for these objects. So here you can see like a stack of fruit that are falling, they are bumping into each other, and the red spots are basically the spots where the bruises will develop. So we can actually simulate the development of bruising during transport. And we are extending this right now to the simulation of tissues to understand how compression causes damage to tissue. Therefore, I have to go to my laptop. So this is a little movie where you can see simulation of a compression of the tissue, and the red is where the cells basically break. This is very recent research that we do. And the idea here is that we want to, if we understand how tissue deforms under force, then we can maybe optimize our handling processes in post-harvest. So this brings me to the end of my lecture. So the complexity of the model depends on its purpose. There are different paradigms for modeling available. Statistical models are the simplest one, but I have a preference to more mechanistic models that really rely on the basic physics behind it. Mechanistic models allow you also to do extrapolations. Statistical models cannot do so. We looked at kinetic models that are based on chemical reaction theory, where you need to have ordinary differential equations to describe what is going on. If there's a spatial dimension as well, then you have to go to more complicated transport models that are typically partial differential equation models that you have to solve using specialized software. And then there are novel developments that try to combine, for example, neural network models with mechanistic models. And with this, I would like to thank you for your attention. Okay, I hope that you enjoyed the lecture.
Okay, thank you very much, Prof, Bart, for the insightful lecture. And unfortunately, due to our limited time, we will need to conclude this lecture part here. And I hope today's session also interesting and valuable for all participants, since our participants coming from wide backgrounds from agriculture, computer-related fields, as well as others backgrounds such as administration, environment, human development. And now we will move to the discussion session. For the participant on site, please feel free to raise your hands if you have questions. And for the online participants, you can use the raise hand feature or type your question in the chat box. And when I invited you to speak, please introduce yourself by mentioning your name and also your country. And now I would be happy to take the first question from the on-site participant first, maybe. Okay. Please.
Good afternoon, Dr. Bart Nicolai. I am Isaac Montefalcon from De La Salle Araneta University, from Philippines. So I realized that the models you were explaining is really useful in gaining understanding how the world works. And I observed that why European countries are so good at excelling is because they understand how the world works through mechanical models. And you know how to formulate the formulas needed to understand how a specific thing works. Like, how can an 300-ton metal fly in the air like that? And the question I'd like to ask is, how are these mechanical models formed aside from having a mathematical background? Say, I am a researcher and I want to understand how how the fruit ripens or how the fruit compresses under pressure when transporting. How do I know, or how do I find the right mechanical model formulas for that?
Well, the nice thing is that everything boils down to the same physics. For example, the airplane that is flying in the sky is governed by Newton's law of gravity and the Newton's laws of motion. But if you have a fruit that is transported in a truck and that is bumping, and then basically the fruit jumps up and down and sideways because of the very same physics. So it's again gravity, and it's basically Newton's law of physics. So if you have like transport phenomena, so airflow in the atmosphere is governed by equations that are called the Navier-Stokes equations. They have been discovered in the beginning of the 19th century by Navier and Stokes. And these equations are the same equations that govern, for example, transport phenomena like transport of gas in a fruit or transport of heat in something that is chilled. And they are the same equations that are used to model the transport of the plasma inside the sun and the universe. So that's the beauty of these models. They rely on physical principles that are universal. This is why you learn them in your physics courses, because they are universal to every process that you can imagine. And then this also means that, for example, if you are interested in studying compression behavior of tissue, then you can go in the literature and look at how they model, for example, compression of steel and of concrete, because you need it if you want to build a bridge over a river. If it's not good enough, then it will collapse, okay? So you just basically have to look around and identify areas which are kind of close to yours, and for sure you are going to find articles and books that cover a topic that is very similar to yours, but maybe in a different setting. Okay, you just have to kind of transfer it to your knowledge. Everything about the biochemistry is actually, it's universal. Eh? You know that respiration is a conserved process. It developed billions of years ago, and you find it in almost every organism, to the most basic microorganisms. There are a few small examples, and viruses don't have a metabolism, but the rest of the biochemistry is universal, and the details change. So you have to accommodate for the small changes in details, but you can rely on all that knowledge that is already available and all those models that have been published and that you can find in textbooks. So it was a general question, so my answer is also a little bit more general, but this is a little bit the idea. You don't have to start from scratch. There's already a lot of knowledge in different areas of science and technology. Okay.
Thank you, Dr. Bart.
Okay, now I will take turns between on-site and online participants. Here in the online participants, I have a question from Miss Danica Mary Aposaga from Philippines. Are you still with us? Maybe you can open your cam so that we can see your face. And she has some questions through the chat box that she find it interesting. This discussion to reduce Postharvest losses using mathematical models, and she's wondering what were the limitations faced by the researcher in the development of the models to assess the respiration rate and or ethylene of the fruits being analyzed.
Oh, there are many limitations. just to give you a simple example, if you do experiments in like a large cool room, which is what we did, these are 200 tons cool rooms, and it's a low oxygen atmosphere. So the oxygen concentration can be like 1%, which means that you cannot enter these rooms because you will suffocate. It's a very painful death, okay? So painless death, so that's good news, but you will be dead anyway. So you either have to wear like a mask, an oxygen mask, or you have to kind of find a way how you can sample the air inside the cool room at different positions because you want to analyze the ethylene. Okay, so this is, for example, a difficulty. Another difficulty is that if you do experiments based on a single jar, there was like a small image of it, I'm not going to go back, but where we put fruits in and then we measure the change of the oxygen concentration inside the jar to estimate the respiration rate, to measure the oxygen consumption rate. The challenge is to make sure that the setup is airtight, that you get not oxygen from outside that gets into this low oxygen atmosphere, because it will jeopardize your measurements. And there are many experimental things. And then the next thing is the model. Because the model that I showed you, there's a much more complex model actually behind it, took the students like almost like a year to develop. And then we were actually traveling to a conference together. And I was looking back at this model and said, Ming, you have to start again. I think it's not good enough. There's a kind of a big mistake here. And he was very unhappy and he tried to convince me that he was right. I said, no, this is wrong. And then he had to start all over. But because he was now very familiar with it, it took him actually just one flight from the United States to Belgium to come up with a concept that was much better than before. And it turned out to be a beautiful model from my point of view. So I was very happy with it. And in the end, he was also happy, but in the beginning, he was really mad at me. So it's kind of things that change and you learn so many things. And then in the end, you see that in the beginning, maybe you should have done it better. And then you have to be brave enough to just throw away what you had and start all over again. So if you're doing good science, you have to be prepared to do that. Okay.
Thank you, Dr. Bart. Actually, she's here with us, the one that asked you a question, Miss Danica. Does that answer your questions, or do you still want to have more discussion?
Good afternoon, everyone. I'm Danica from the Philippines, and actually my specialization is more on post-harvest losses, but it's more specific on converting certain food waste into a natural preservative and additive. So I was just curious on certain limitations, and I think the answer of Professor Nicolai is very clear. So, in case we are going into that specific research field, we can take that into consideration. So again, it's actually a very good topic. It's very informative. So again, thank you, Professor, for the answer and also for the discussion.
Okay, pleasure. Thank you.
Okay, thank you. Now we can move to the on-site participants here. Okay, you can ask your questions.
Hello, good afternoon, Dr. Nicolai. I'm Jose. I'm from the Philippines, from the LaSalle Araneta University, and I have two questions for you. First is, your AI data models, or your data models, do you host them locally in your school or university or facility, or do you outsource that work to companies like OpenAI and Anthropic? And my second question is, how much data do you gather in order to train these AI data models?
Yeah, that's a very good point. We use a particular type of AI models because we use AI to interpret, for example, RGB or X-ray images. And the models are called convolutional neural networks. These are neural networks that are used for finding patterns in images. Okay, for example, you have many of these softwares where you, for example, see a bounding box around the head so that identify persons and so on. That's a kind of an AI topology that we, for example, it's called YOLO, but there are many different topologies for doing such image analysis. And what we typically do is we run the, well, we have special servers in my lab to run these models, to train the models actually, because this is where all the work goes into. Or if it's becoming too big, then we basically go to the supercomputer, the Flemish supercomputer, to train them. It depends on the size of the models and the size of the data sets. So we typically have models that are of the order of magnitude of about 1 million weights that we have to train, which are still much smaller than the big transformer type of networks that are used for things like ChatGPT or Claude or things like that. So we basically train them in-house. But what we do is we use existing architectures and we kind of tune them a little bit, maybe add another filter there and then remove something else or add another layer. But we often use pre-trained network, as they are called. Okay, so there are also large internet bases of millions of images that you can use to train your networks yourself, but you can also use pre-trained networks that are already trained. And maybe if these networks can distinguish like a cat from a dog, from a human, from a bicycle, maybe they also okay with distinguishing like a cavity in a pear from a pear that does not have a cavity. So we basically use often these trained networks and then we train them again to kind of fine tune them, or we use a subset of the network. So this is how we work. But we do it all in-house. Yeah.
Thank you.
Okay, thank you. Is there any more that wants to ask questions? Maybe from the Indonesian participants? Okay, so we have from the online participants here. Okay, from the Gadjah, you can open your cam so that we can see you. Hi, Dr. Nicolai. Thank you for the informative topic. In measuring oxygen concentration in fruit, where is the sensor usually inserted? Thank you.
That's a very good question. So measuring oxygen concentrations in fruit is very difficult. Actually, we have been looking for methods for many years, and the only way that is, in my opinion, well, that has been published about so far, is by using needle electrodes. And the needle electrodes, you basically insert the electrode through the skin of the fruit into the parenchyma tissue to the center where you typically want to measure the concentration. But the problem is that, well, these sensors are usually developed for softer tissues or for very small distances, like we even have microsensors and so on, to measure, for example, the oxygen concentration in like seeds or in leaves and so on. But if you want to go to the center of a fruit, which is like a bulky object, then they need to be long, and then typically they would break halfway. So that's a problem, the mechanical strength of the sensors itself. Another problem is that you can have leaking of oxygen along the shaft of the sensor. And then you don't know what you are measuring. Are you measuring the oxygen concentration in the fruit, or is the mixture of the oxygen that seeps in along the shaft from the environment? So it's very difficult. But so far, there are not really standard alternatives. So there are some ideas that we have and so that people tried, but there are, for example, in related sciences where they look at, we worked with a group in Italy, and they look at oxygen profiles in meristems, but meristems are very small. And what they did is they developed what they call biosensors. So it's basically like a sequence, a gene sequence of like a dye, and there's a promoter that can be activated that depends on the oxygen concentration, for example. And the whole idea is that they have this formed or meristreams of transformed plants. And then if they reduce the oxygen, then the color changes. And then they measure the color. But this is only possible for very, very thin slices or very small tissues like meristems. But for bulky organs, there's not really an alternative so far. Yeah.
So, thank you very much for that explanation. It's much clearer. So based on your answer, Doc Nicolai, I assume you only put one sensor in a fruit, right? Not like 2 or 3 sensors in a fruit.
Yeah, well, basically most of the work that we do is do the modeling. That's to avoid the need for sensors. But of course, every model that you have should be validated. And we have validated this by these needle electrodes, but they are typically mounted into like into an arm that you can move. So we basically push the sensor inside the fruit, we measure the oxygen concentration, then we push it a little bit further, measure the oxygen concentration again and again and again, so that we get at least like a one-dimensional oxygen concentration profile in the fruit.
So may I ask what tropical fruit have you used in your research with regards to oxygen concentration measurement? What tropical fruit?
Well, we have several projects on tropical fruit, but we have, let me think now. I don't think we have, well, you do remember that we do any tropical fruit in terms of gas transport? I don't think so.
Gas transport.
Does
Also
tomato,
not yet in fruits.
does tomato work? Tomato is subtropical of origin. So we had like 2 years ago, we had a publication, Plant Physiology, on oxygen profiles in tomato fruit. Not really a tropical fruit, but subtropical, maybe.
One last follow-up question. If I understand it right, you measured the oxygen concentration to check the ripening of the fruit, right? Or of the meat. So on the other hand, I think one of your presentation, you measured ethylene concentration as well?
You mean in general, like in practice, whether we measure ethylene concentration or what is exactly your question?
Yes, in the model,
Yeah, in the model,
what are the parameters?
Yeah, so in this particular experiment, actually, I showed you only a very small part of the data, but we measure in different types of controlled atmosphere conditions, so DCA conditions, normal controlled atmosphere conditions, and also air. And then typically what we measure is the oxygen concentration or the oxygen consumption rate, the carbon dioxide concentration or the carbon dioxide consumption rate, and then the ethylene production rate. Yes, and we measure ethylene. We measure typically these gases with gas chromatography. We have like small compact GCs that can measure these gases.
Okay, so I have to ask that question because I was curious to check if which parameter is better or much better to determine the ripening or browning of the fruit. Is it the oxygen parameter or the ethylene parameter or what is this totality, complementary of all the 3 parameters that you have mentioned?
Well, if you really want to measure ripening, then the best thing to measure is the change of the ethylene evolution rate. Because that's the most direct indication of the climactic rise, and typically you have to pick your fruit just at the start of the climactic rise, not earlier, because then they will never ever ripen, or also not later. But the thing is that in practical conditions, it's very difficult to do because it's a gas exchange method. So you need to take fruit, put them in jars, and you maybe can only do like 8 jars. And then you have to measure at 2 different times with some time in between, maybe an hour, maybe a couple of hours and so on. That is really a lot of work. And so, for example, my lab is also responsible for determining the picking date, so the ripeness of fruit in whole Flanders in Belgium. that is totally impossible to do. So this is why we measure so-called maturity indicators, which means we measure the evolution a couple of weeks before or months before harvest of some properties that we know that are related to ripening. For example, the sugar content or the starch content or the acidity or the firmness or the color changes. And because all these properties, they change during the ripening process, if you measure some of them, you can kind of find another relationship. And for that, we have actually a verbal model. We don't have a quantitative model, but we're working on it with ripening. And then we give an advice to the farmers. And then we would say, well, you have to pick your apple fruit between September the 15th and September the 23rd, for example. So the direct gas exchange measurements, which are the most direct ones, especially ethylene, but oxygen consumption rate is almost equivalent, are the best, but in practice, it is just not possible because it's too much work.
Okay. Thank you very much. Yes.
Thank you for your question.
I learned a lot. Thank you very much.
Thanks.
Okay, thank you very much for Ms. Jezebel. And due to our constraints, we have to wrap up these sessions. So thank you very much, Prof, Bart, for this insightful lecture today. And I have to wrap up this session by giving a conclusion from your lecture. So we have gained much clearer understanding about the modeling that is not merely mathematical exercise, something like that, but a powerful scientific tool used to analyze post-harvest process to explain what happens inside biological systems and also to support better decision in cold chain management. We also learned that from different modeling approaches serve different approaches and that the choice of model should be always depend on the question what we want to answer. And one of the most important insights from your today's lecture is the distinction between the statistical and mechanistic modeling, and also the statistical models help us to describe patterns in data, and also for the mechanistical models allow us to go deeper by linking between the physical, chemical, and also biological principles. And in that sense, modeling becomes not only a way to fit the data, but also a way better understand nature and improve real systems. So the examples shared today, ranging from the climateric ripening and also gas transport in fruit to computational fluid dynamics in cold rooms and to discrete element from modeling transport damage, clearly show how modeling can be applied across different scales of post-harvest chain. And overall, today's session has shown us how science and engineering can work together to reduce food loss and also to improve refrigeration efficiency and also to support more sustainable post-harvest system. Prof, thank you once again for your time and for your generosity sharing your expertise here and for being with us today.
Pleasure. Thank you.
So thank you. And I would also like to thank all the participants, both on-site and online participant here for your presence and active engagement. And also I would like to thank one of our lecturer here for attending our class here. On the back, we have Dr. Bayu Nugraha. He is one of the students of Prof. Bart in KU Leuven. Thank you for joining us today. And since we all here still on day 2 of this short course, I hope today's session has given you more motivation and curiosity to continue joining us for the upcoming session. And also we still have many valuable discussion ahead. And I sincerely encourage all of you to stay engaged and make the most of these learning opportunities. With that, I return to MC today, and thank you for having me as your moderator, everyone. Have a wonderful day.
Okay, thank you so much to Professor Bart Nicolai for being an insightful lecturer about biofluidics in post-harvest and cold chain, cutting food loss and climate footprint. We would like to give the appreciation certificate that will be given symbolized by our moderator. And also, we would like to thank Ms. Hilda Maya Cynthia Dewi, who led the discussion. Before and the certificate of appreciation will be given by the Vice Chairman of a short course 2026, Ms. Taliana Siha, MSc MBA. With the station of appreciation certificate already implemented, then the third material of this summer course has ended. For operators, you may stop the recording. Thank you. We